Professor, you've been talking about various kinds of ways to forecast traits of human beings. You say some traits can be uh, forecast if you know the behavior of one gene. Some traits like intelligence, you said, may include 100 genes or a large number. Uh, and we go in and manipulate. We can go in and manipulate them. But is it true that manipulating 100 genes, like with intelligence, is much harder than manipulating a single gene, and perhaps even more dangerous? Is that true?
Well, big brother, seeing how it's 100 genes for intelligence for you. In my case, it might be 1,000. Yeah, it's a lot tougher. It's a lot tougher. And here's why. Because we use a new little tool. I love tools. I'm a science nerd. I like these brand new cutting edge tools. And this is not, 5 horsepower, impact drill. This is a little bitty molecular scissor that can go in and cut DNA. And it can read the DNA. Your DNA is pretty boring. It only has 4-letter codes, but they're all stretched out. And if you have a set of, let's say, roughly 20 letters in all their combination in a row, it can see that, it recognizes it, and it can make a cut.
So what we're talking about is on inside of gene, going inside the gene itself and snipping and sewing and redesigning the gene itself.
Right.
What we're not talking about at this stage is taking an entire gene and replacing that gene with another gene, say from a fish or avocado or something.
Okay, no, but we could do that. And so when this little scissor cuts the DNA,
yeah,
the cell says, "Oh my goodness, my DNA has been cut, fix it." And so it has a whole machine inside that tries to fix the cut. Okay? Now we can trick it. We can trick it so that it doesn't fix it properly. And by doing that, you've totally screwed up the gene. Okay? That's how you can, as we say, knock out a gene.
Oh, really?
Yeah. Or we can do what you just suggested, and that says, okay, let's make 2 cuts. Or let's make a cut in a way, and then we add some DNA. And when the cell fixes the DNA, it inadvertently switches out the DNA that you put in for the one that got cut out.
Oh, man.
Isn't that cool?
Okay, and so if you have a trait that exists on one gene, like the AIDS virus is our example, then that's reasonably straightforward, if not easy. It's straightforward
that was an easy one
to correct the gene, and so that gene makes a correct protein that is immune to HIV virus.
Well, wait a minute.
But
I think we're confusing folks a little bit by saying it that way. So let me.
Yeah. Okay.
All right. To say that a gene is correct or not is a little misleading, right? So some people have one version of a gene that is missing a little bit, and those people who have that gene, that form, that variant of that gene, they're resistant to HIV.
Yeah. Okay.
But you can't say that's the correct form, right? We don't actually know whether maybe that those people who are resistant to HIV, because their form of that gene lacks, the part you need that the virus recognizes, maybe they have some problem we don't know about. Right? And that's part of the ethical story that maybe you want to address later on, is that We think we know what we're doing at any one time, but there's the unknown out there. Okay.
Okay. So I get your point. Even when we're manipulating one gene, there's some uncertainty that we need to be concerned with. And this uncertainty, if I'm getting this correct, is just multiplied when we start manipulating more than one gene.
The technical part gets tricky because
Yeah.
when these scissors come in and they cut the DNA, they do so with a certain efficiency. Let's say I'm going to make something up, 10%. Okay. Now that's 10% for that gene. So you would have to screen like more than 10 embryos, right, to find one in which you have the engineering was correctly done. All right. Now let's say you had 2 genes and each one is operating at 10%. You're the mathematician. What is that? That's like an exponential thing, right?
Well,
now you're looking at 1 in 100.
It's a square. You have to have 100 embryos in order to find at least 100 embryos.
That's right. So you see, that's the technical thing. That's the technical part. Now, every day we're getting better and better and better. And we're approaching 100% in some cases. That's good. So, but these are just engineering problems that we're going to solve. They're not the ethical ones. These are engineering problems, and I think nothing like technical like this is going to get in the way of this happening.
Well, no, you've hinted at an ethical problem in your description. If one gene requires 10 embryos and you pick out the one you want, that's right. That means 9 embryos have to be disposed of.
Right.
If it takes 2 genes, each with a probability of 10% of getting the correct, no, sorry, I'm using the word correct, but getting the desired
form
outcome, then you have to screen 100, at least 100.
Yep.
To find the one you want, and that means 99 embryos get flushed.
Yep.
Okay, so that is certainly an ethical problem we have to think about.
Yep, afraid so.
Okay. Well, that's fascinating. I guess that's it for today. This is Click.
It's Clack.
Signing off. See you next time.
Goodbye.